Hi everybody. In this series of videos we just want to talk about delivering the nucleic acids into the cells, such as the nucleus or only the cytoplasm of the cells. Well, let's have a look at the structure of a cell for those who cannot remember. You see here that inside this cellular membrane, this is the cellular membrane of the cell, which is negatively charged. Remember this. And here we have the nucleus, in which the DNAs are stored, the genetic information of the cells. You see this purple around, this is the cytoplasm of the cells. Well, when the DNAs and RNAs, these two nucleic acids, are not working efficiently or properly, there will be some diseases originated from them. Let's have a look at the difference between the DNA and RNA. As you see here, the DNA are the double-stranded molecules, and they are responsible for storing the information. On the other hand, the RNA molecules are the single-stranded ones, and they are responsible for getting the genetic codes of these DNA molecules to express a certain type of the protein. Well, and as I mentioned, these molecules are negatively charged, so it will help us to encapsulate these negatively charged molecules inside a positively charged complexing material that I will mention in about 10 minutes. Just remember, when I mix the DNAs or RNAs with, for example, positively charged polymers, I will have a polyplex. Polyplex means a polymer which inside has the nucleic acids which are negatively charged. For example, if instead of the, let's say, polymers, I use the lipids, the liposomes, to encapsulate these nucleic acids, I will have the lipoplexes. Anyway, we will mention them in the following slides. As you see here, the messenger RNA is responsible for expression or the synthesis of a certain type of protein. How does it work? Here in the cell we have the DNA, in the nucleus we have the DNA which will synthesize a mature mRNA, messenger RNA, and it will cross the nucleus membrane to come out into the cytoplasm. So let me show you again here. So inside the cell, the DNAs are going to work, and then the messenger RNA, it will come out, and inside this purple cytoplasm it will continue to work for the expression of the proteins. How does it work? Here is the mechanism for the synthesis of the a certain sequence of the nucleic acids. And because you know the proteins are just the combination of different sequences of the nucleic acids. So how does it work? Well, the sequence at which different nucleic acids have to be connected to each other is really important to produce that certain type of protein. But who is responsible to tell us what is this sequence? The messenger RNA is responsible for that. Where did the messenger RNA gets the information about this sequence. The mRNA got the information from the DNA, and we told you that the DNA is responsible for storing the information. So it has all the genetic data of the cell inside that. A mature messenger RNA, which is a copy of this, will be formed, and it comes out, and by the help of this messenger RNA, through a transcription process, this process is called a transcription, that the ribosome will help the mRNA and these tRNAs to be connected to each other to help for the formation of a certain sequence of the amino acids. So just remember that this process is happening outside the nucleus. Sometimes we can interfere, we can block this transcription process for the expression of some cells. But sometimes we have to go inside the cells to change the, let's say, genetic codes to introduce something inside. Well, in gene therapy, we can introduce the nucleic acids into the cells to do what? To fight against the disease gene and to change some behavior of the cell. For example, in the matter of cancer cells, we know that the cells are infected and they are, let's say, proliferating more and more and more. So something is wrong. The genetic code of the cells have been changed. So if we treat the cancer cells genetically by delivering some genes, we can, let's say, stop the uncontrolled proliferation of the cells of the cancerous tumor. The process in which we deliver the gene for gene therapy, in science it is called transfection. Let me type it for you. Transfection. So it means to delivering the genes. Okay, let's continue with these slides. Well, for example, in a certain type of disease, if the specific type of protein is not expressed, I'm sorry, I received a message from my friend, I have to close it. Okay. Well, as I was saying, if we have a disease in which a specific type of protein is not expressed, we can deliver the genes to help the genetic code to be modified in order to produce the proteins again. However, if you look at this diagram, you see that most of the studies are related to cancer diseases. And how does it work? Well, we want to deliver a type of gene into the cancer cells in order to trigger the cell behavior for destroying themselves. It means that we want the cancer cells to undergo a suicide. So by doing that, we can, let's say, stop the growth of the cancerous tumor. On the other hand, we have these the application of gene therapy in a so-called antisense, in which we will interfere with the mRNA outside the nucleus to block the expression of a specific cell. If you remember here, I told you that outside the nucleus, within a so-called transcription process, the specific sequence of the nucleic acids will be formed to form the proteins. So in this so-called antisense, we can interfere with this process to block the production of the proteins, to block the expression of specific proteins. Well, there are two different main ways in order to deliver the nucleic acids into the cells. One is the direct delivery, in which that we design a nanocarrier. A nanocarrier can be either a nanoparticle, such as a polymeric nanoparticle which contains the nucleic acids. I told you, if we use, for example, the polymers to encapsulate the nucleic acids such as DNA or RNA, they are called the polyplexes. If we use the liposomes, they are called the lipoplexes. But these nanocarriers are not necessarily made by the humans. We can also use the viruses, because the viruses are made by the nature and they are much more efficient than what the humans produce. The second strategy to deliver the DNAs and RNAs or nucleic acids into the body is the ex vivo. As you see here, we will isolate the unhealthy cells at the beginning, and then we can apply a sort of an electrostatic field to make these cells more permeable, to open the cellular membranes, pores more and more, so they become more permeable, and the nanocarriers can enter the cells to treat them, to deliver the new nucleic acids, and after they are genetically treated, we can implant them again back into the body. Well, I'm sorry, something happened with my system. Well, let's talk a little bit about the viruses now. As we mentioned just some seconds ago, the viruses are much more efficient compared to the nanoparticles, the artificial nanocarriers that we are going to synthesize. The fact is that the viruses are able to replicate. Replicate means to create a copy of themselves. So the viruses are able to replicate. But the problem with this is that let me just tell you what is the replication, how is replication useful for us. You know, the replication of the viruses will increase the efficiency of delivering the nucleic acids inside the cells. On the other hand, if a lot of viruses try to replicate, the problem is that the immune response of the body will be activated, and this is something that we don't want to happen. Well, so we have to disable their ability for replication. Okay, we have to hamper this ability for replication in order to avoid the strong immune response of the body. Well, if we do this, there is one problem with disabling or deactivating the replication ability of the viruses. If we do this, only the cells, the cancer cells on the outer shell, can be treated by the genetic codes that the viruses introduce inside the cells. And the other cells, which are in the inner shell, are not going to be treated. Well, this decreases the efficiency of the viruses. Just imagine, on one hand, if the viruses can replicate, they are going to be more efficient, but the immune response of the body is going to be activated. On the other hand, if we decrease the replication of the viruses, only the outer shell is going to be attacked, so they are going to be less efficient. So as a matter of fact, there should be a compromise between these two issues. On one hand, we want to minimize the immune response of the body. On the other hand, we want to maximize the replication to make the viruses more efficient. Well, the problem of the immune response of the body is so, let's say, important because it can lead to the death of a person. For example, there was a study in 1999 in which a person died when he was suffering from this problem, and he was undergone but by the viral vectors to deliver the nucleic acids. And when the immune response of the body was activated and triggered, it was so massive that led to the death of that person. In this slide, we have 5 different types of viral vectors that I don't know whether you have to memorize all of them or not, but just remember that the retroviruses, these viruses are dangerous because they can enter the cells when they are dividing, because when the cells are dividing, they're porous. The cellular membrane is going to be more porous so that the virals can lead to the nucleus. We have the lentiviruses. These viruses are even more dangerous because even when the cells are not dividing, they can enter the cells. But we have these adeno associated viruses, AAV, which do not cause a strong, let's say, immune response of the body. They cause a very mild immune response of the body. So these are the ones which are appropriate for delivering the nucleic acids into the body. But there is one limitation with them, is that they are so small. So the length of genetic code that can be carried by these viruses will be limited. So here we have a summary of the limitations that we mentioned. Well, the limited tropism of viral vectors. Well, the problem with this is that the viruses are not so selective towards only a specific type of cell. They can bind, they can enter even other types of cells. So the selectivity of these viruses is not so strong. The duration of gene therapy, for example, the gene therapy can work for some time, and after that the cells are not gonna be affected by that virus. Also, we mentioned about the immune response of the body, and let's say the last one, integration is not controlled. Well, it says that the viruses can enter the nucleus, and when they enter the nucleus, they can integrate with the DNA inside the nucleus and lead to the trigger of the growth of cancerous cells. So the cells will develop the tumors if they integrate into the nucleus with the DNAs which are already inside the nucleus. Well, because of all these limitations that we mentioned about the viruses, we have to use the non-viral gene delivery carriers for delivering the DNAs or RNAs. And I see that I have already talked about 22 minutes, so let's continue in another video.